modern SNP genotyping arrays measure your genotype at about 1 million locations, and then through imputation, we can get to about 10 million variants per individual. And the cost of that for the array itself, actually, if you buy them in volume, can be down to about $10. And so the all-in costs, including the chemical reagents and stuff, are well below $50. So UK Biobank reported genotyping costs of about $30 per individual for their sample. So you're talking about differentials here of 30x in cost. So at fixed budget for the kind of work that I do, it is much, much more efficient to do array-based genotyping of a very large population rather than deep sequencing of a much smaller population. Now, that deep sequencing is important if what you're worried about is rare variants, perhaps of very high impact, whereas here, because of the polygenic architectures that we're looking for, what we really want is many, many different individuals, and we just want not a perfect genotype for each person, but 1 million SNPs called or 10 million SNPs called from each individual is enough for us to do our work. So the basic methodology here is that we are given in a typical analysis some hundreds of thousands of individuals, and we have some phenotype information about each of the individuals. Now, in the case of disease, which is what I'm actually going to focus on for most of my talk, the situation is typically that we only have a few tens of thousands of cases. So those are people who actually have the disease. Like, say, you have 30,000 people with type 1 diabetes, but then you have 200,000 or 300,000 controls, who are people who don't have diabetes. And what you're doing is you're showing those examples to a learning algorithm. I'll give you some details about those learning algorithms if there's time later in the talk. But they go under the heading of sparse learning. And the reason, what we mean by sparse learning is that the number of candidate features which might be used to predict the phenotype is of order of 1 million. So say you have 1 million reads from the array. But it turns out even for the most complicated phenotypes, we don't need more than a few tens of thousands of individual SNPs. So most of those 1 million SNPs actually are not activated in the predictor and don't need to be activated in the predictor. It's about maybe 30,000 or 1,000 that we need. So that's what we call a sparse learning problem, because most of the features that are shown to the algorithm are not actually useful. Now, to publish results in this field now, you have to validate out of sample. So if you look at the general level of rigor for biomedical research or biological research, I would say this falls on the very high end, because a typical situation would be we train on this big data set, we freeze the predictor as an outcome of that training, but then before you can publish a paper, you must find other populations of people, people who grew up in different decades, people who grew up on different continents, and then you must test the predictor on that validation set. So you must show that, for example, the risk prediction or the phenotype prediction in the validation set is comparable or similar to what you got on your training set. And so you're showing that, you're at least increasing the likelihood that the effects that you're discovering are robust genetic effects because they replicate in different populations that have experienced different environments. Now, what's actually going on now, I'll say a little bit about it further in the talk, is large-scale tests of clinical utility. So, for example, we have very good polygenic breast cancer risk predictors now, not dependent on BRCA1, so people who are negative for all the BRCA variants, but we look at 1,000 other locations on their genomes and determine, for example, the subset of the population that's at high risk. And there are now EU-funded medical trials in which they're looking at things like allocating mammogram resources to that population earlier than for low-risk populations. So that's really where the research is now. Okay, this graph shows the sort of very rapid increase in the number of papers constructing polygenic risk scores using procedure like what I just described on the last slide. And you can see it's really just taken off. A few years ago, any polygenic risk score that was constructed was just kind of a curiosity. It was sort of like, I'm doing this big GWAS, I got a bunch of hits. Well, just for fun, let me see how much of the variance I can predict in the condition, phenotype using the SNPs that I discovered. And it was just for fun. It was not useful at all. But in the last year or two now, we have actual predictors that work. And so hence the clinical investigations that I described. Okay, so I predict that, and again, so this is what I meant by most of the talk is actually nothing to do with embryos. In this case, on this slide, what I'm talking about is our healthcare system or the healthcare system in some other country like the UK. Routine clinical use of genomic data of the kind that I'm discussing here will arrive relatively soon. we have good predictors for a number of very, very impactful common disease conditions. And while we can see that the quality of these predictors is improving in real time as we get more data, they are already good enough that for outliers, there are clinically actionable outcomes. So in other words, if you could genotype a large population of people and you just look at, say, the people who are top 1% or top 2% risk, the absolute risk that subpopulation has for having the condition is high enough that there are clinical actions that should be considered. And so you will see, I believe, eventually inexpensive genotyping becomes standard of care in most healthcare systems. Now, whether that takes just a few years or longer depends on all kinds of institutional and societal things that are hard to predict. Okay, so here's an example of some results of the type that I've been discussing. So, sorry. This is coronary artery disease. This is type 2 diabetes. This is breast cancer risk. And always on these plots, what is plotted here on the horizontal axis is the percentile of the individual or a class of individuals on the polygenic score. So these would be all the individuals, say, that have 99th percentile polygenic score. These are all the individuals, say, that have 50th percentile polygenic score. All of the dots are replication data. So this is not the training data. This is, you train the predictor, you freeze it, then you bring it to another population, then you check how well it does in the other populations. And you have a kind of characteristic nonlinear behavior here. This is the risk elevation. So the increased risk that individuals, conditional on that polygenic score, the increased risk that individual has. So you can see there are many predictors now where people who are in the top percent or a few percent of risk score are going to have 10 times, even 15, 20 times the risk of an ordinary individual for having the condition. And so it's these people that would be selected out for additional clinical intervention or early screening, depending on the disease condition.
Stephen, sorry, just to be clear, for example, breast cancer, this is not the beautiful outlier on the right. This person has an enormously elevated, this group of people, enormously elevated breast cancer. And they happen to have hit the negative genetic jackpot.
Right. So
they have all the benefits. But is that BRCA1 and BRCA?
Yeah. So in the case of breast cancer, these are people who are BRCA negative. So the risk here is being induced only by SNPs that were not well known just a few years ago, and they're scattered throughout the genome. So typically, I think the best breast cancer predictors now use between 1 and 5,000 different SNPs, none of which are BRCA. So obviously, if you know you have the BRCA variant, that's one thing, but this is a different thing. Also, these dots are not one person, they're many people, because you actually have to have enough people in that bin to compute a probability. So for some reason, these people don't use error bars. I'll show you some results like this that actually have error bars. And all of these now have been validated by multiple groups. So we have predictors very similar to the ones that were produced. This is a group at the Broad. This is Kara et al. Sorry, having trouble with your pointer. Yeah, so this is a group at the Broad. Now, in addition to disease risk, of course, there are quantitative phenotypes that we can try to predict. So this is a result from my group. Height is a kind of classical genetic trait, and it's known to be highly heritable. There are sort of classical studies like twin studies, adoption studies, where you can get some estimate for the heritability of height. Now, what we've done here is train on about I think, almost 500,000 people. And the predictor produces something which has accuracy of about 1 inch. So this is a validation set, not the training set. These are women in red, men in blue. The predicted height in centimeters is given here. So these are all the people that are predicted by the algorithm by the predictor to have height of 180 centimeters. And if I look at all the people in that bin that have predicted height 180 centimeters, the standard error in the distribution of those people is about 1 inch. So in other words, if we discover DNA on a gun at a crime scene, and we could then genotype that DNA and run the predictor, we could say the guy who shot this gun was 180 centimeters plus or minus 3 centimeters, standard error. And that was not possible a few years ago, but it is possible now to do that. This is the genetic architecture for human height. If I were to lay out your 23 chromosomes and put a coordinate system on them from 0 to 3 billion base pairs, these are the locations of the 25,000 or so SNPs that are used in the predictor. If the black line goes up, it means having the rare variant at that location makes you taller. If the black line goes down, it means having the rare variant at that location makes you shorter. And the predictor has to sum up all of these effects in order to make these predictions. Okay, but this is currently the best known polygenic predictor of a quantitative trait that we have. Yes?
Now you could also go back and start dropping out in individually.
Yes.
And you ask, what's the minimal set I need?
Yes. Yes. That's a great question. And when I talk more about the algorithms used here, the algorithm has already done what you described. So in a way, this set of 25,000 can't really be made smaller without losing predictive power. It can be made larger without gaining predictive power, basically. So, okay.
For a particular individual, can it say this is the gene that most impacted them being predicted?
Not really, no. It's the way this stuff works is you're training on this big population, you freeze the predictor, and then on a given individual, you might find significant errors. So you might find someone who's predicted to be reasonably tall, but they're actually quite a bit shorter, but that person's an outlier, and almost all the people lie like within a few centimeters of the prediction. And of course, the comment about environments here, these are all people who have lived in first world environments. So the training set is largely British people from UK Biobank. Validation sets we've looked at have been Americans, New Zealanders, people from other parts of Europe. Okay, so in a paper that we completed at the end of last year, we constructed predictors for all of these disease conditions, and all of which were validated out of sample. This curve illustrates the characteristic nonlinear behavior on all of these curves, which is on all of these plots, which is that as you go toward the outliers, the tail of the distribution in the percentile risk and the percentile score, you get this very fast accelerated growth in the actual odds ratio and the actual risk that that person actually has the condition. And it turns out this red line, this is stuff that theoretical physicists love, this red line can be computed using a simple formula. So we've actually derived a simple formula that produces this red line. And these black dots with error bars are the actual data from a validation set. This is atrial fibrillation, so it's an irregularity in your heartbeat. Our ability to predict risk for this, as you can see from the fit of the points. So we didn't fit the red line to the points. We generated the red line with a single formula, and then we laid the data down on the red line. So the theory, I think, is very strong. And so we could even say that as you get into like 99.5 or 99.8 percentile in the population, your risk really is going up enormously. Okay, here's another validation. This was just published, I think, in the last month or so. This is a validation of the coronary artery disease predictor. It was done in Quebec. So they have 11,000 people in this sample, all French Canadians. When this came out, there was a little blurb in the press from the American Heart Association. PRS, polygenic risk score, is one of the biggest advances in heart disease and stroke research in recent years. So what's shown here is two different predictors, which are they're slightly different, but they're highly correlated with, but they're highly correlated with each other. And the dots, the green dots, are people who do not exhibit coronary artery disease. So people who have low risk scores are very unlikely to have coronary artery disease. But if you have a high risk score, you're very likely to have coronary artery disease. And again, just to repeat myself, the predictor was trained in this case for coronary artery disease. In our case, we did it with British people. In, I think, Kara's case, it was maybe mostly Americans. But this is an out-of-sample validation on a somewhat different ethnic group who also live in a different country. So this is typical of the kind of replication results that you find. Now, this plot is a little bit misleading because you can't see the density of green dots. So you might say, oh, it doesn't look like the probability of having CAD is actually increasing that strongly. But you can't see how many green dots there really are once the space is filled in, they sort of pile up on each other. So what is true is that these people are very unlikely. The conditional probability for people down here that they have CAD is very low, and for here it's very high. Okay, so how is this going to be used in the healthcare system? So we have about 20 or so common important disease risks for which we have this kind of level of performance now for predictors. And so you can imagine two kind of obvious things. You take some threshold to define what the outliers are, high-risk outliers. And you can imagine lifestyle interventions. So if someone's high risk for type 2 diabetes, you can imagine that somehow you try to get them to exercise and lose some weight, et cetera, et cetera. Maybe, and there are big trials now underway to test this hypothesis, maybe if someone is told they have some genetic predisposition for the condition, they're more likely to comply and adhere to these recommendations. Maybe not. We just don't know. It's an aspect of human nature human behavior that we don't really understand. I think more practical is something like this. So in the case of, breast cancer, there already is a standard of care for people who have the Mendelian mutations which predispose. So if you have BRCA, there's already a standard of care where women who have BRCA variants typically will start getting their mammograms about 10 years earlier, so starting in their 30s, and they'll have them more frequently. And that's found to be, I guess, an effective way to deal with that increased risk. Once it's widely understood that we can identify women using the polygenic predictor, that same standard of care might be applied to that new population. I want to emphasize that the new population is 10 times as big. So in the general population, the fraction of women that have a dangerous BRCA variant is only at the sort of 1 in 1,000 level. But we can identify women who are in the top 1% for polygenic risk who have comparable risk for breast cancer, who are not carriers of BRCA. So there's a 10 times larger population of women walking around unidentified today who maybe should be treated the same way medically as women who have the BRCA variant. And so that's a kind of clearly important bit of progress for medical science. What's interesting is that if you make some crude estimates of the benefits you get from population-wide genotyping, it seems to me that already the benefits outweigh the cost of genotyping for the whole population. And I think on this slide, I say something about that. So you could do a cost-benefit analysis of the following kind. Imagine that you have to pay G dollars per individual to do this inexpensive genotyping. So imagine you have to pay $50 or $100 to genotype each person. And then you can ask, conditional on some threshold, like top 1% risk, you can ask, how much money do I save by knowing who these high-risk people are ahead of time? So some fraction of them will actually develop the disease, but they'll get some benefit from having been screened early. And then there's a cost for the screening. So there's a little formula you can write down. And in this formula, you need to sum over all the disease conditions. As I said, there are about 20 of them now, for which you can do some level of polygenic risk prediction. If you plug in numbers for just breast cancer alone, it looks like, again, this is just a naive kind of physicist analysis. This is something much stronger should be done by real medical economists or policymakers. It looks like you break even. It looks like the cost of genotyping all the women in the United States is about paid for by what you save from detecting breast cancer early in that 1% that are currently walking around at high risk without knowing it. So as a public health proposal, I would say population level genotyping already makes sense, and it will only make more sense as these predictors improve in quality. Okay, so now for those of you familiar with the UK, they have something called the NHS, which is a national healthcare system, and they already have plans to offer out-of-pocket genotyping. So in other words, they've built a whole infrastructure for genotyping within their system, and now if you come in and you say, hey, on this next blood draw for my checkup, I would like a whole genome done, or maybe an array genotype, and I will pay you to do it, then the NHS will process that for you. And eventually, I think the data will get back to your doctor. At a speech to the Royal Society in March, the current health minister in the UK described his own polygenic risk scores, and he proposed that the UK move to this kind of population level genotyping. So it's possible that this kind of thing could become standard of care, at least in some single payer systems. In the US, it's much more complicated because we don't know who's going to pay for it. We don't know who's going to benefit from it. If you get genotyped by your current, say, Kaiser, but then you move to Michigan and then you have a different healthcare plan, maybe that second system gets all the benefits from the information, but Kaiser paid for the genotyping. So in our system, which is not single payer, everything is much more complicated. Okay, now I'm finally to the point that Fyodor advertised. So does anybody know what this is? Blastocyst? Embryo, yes. So these are embryos. Currently, worldwide, there are about 1 million embryo genetic tests done each year. And the basic procedure is the following. So current standard of care at good IVF clinics is that the embryos are frozen in liquid nitrogen. And the main reason for that is to give the mother-to-be's body time to recover from the hormone shots that are required to overstimulate egg production. And so because that's the standard of care, and so the embryos might be frozen for a month or something like that, there's a unique moment where you can take a little biopsy off the embryo right before it's frozen. It's about 100 cells, and then you take the biopsy, and then you freeze it, and then you have about a month or whatever time the elapsed time is that they're frozen to actually do all the genetic testing and report back to the physician, et cetera, et cetera. Now, these million tests are almost all for aneuploidy. It's less common for people to screen against Mendelian variants, but that is also done. Now, what we've done in our startup is build a molecular pathway which allows the standard biopsy to then be amplified. And then we deliver both the aneuploidy report, a report on hundreds of common Mendelians, the most dangerous Mendelians. And then we also report these common disease risk scores for each embryo. And one innovative thing we do is that we actually genotype, we take saliva from both parents and we use the haploblocks that come from the parents to error correct the genotype that comes for the embryo. And we actually get higher accuracy on our embryo genotype now than you would get, say, from 23andMe from a commercial genotype. So it's very accurate. I think 99.6 concordance, 99.6% concordance. And the cost is currently, I think the actual pricing is something like $1,000 case fee and $400 per embryo. And we currently have about, I think, 15 IVF clinics in, I don't know, about 5 or 6 countries sending us samples. So this is actually something that's real. Okay, and this is a typical report. This is a circus plot, and if there were a problem like a Mendelian, it would be indicated in red. This is a list of the common disease risks that are reported on in the report. The report goes back to the physician. It doesn't go back to the parents. It's advisory only to the IVF physician and the genetic counselor at the clinic, and then they decide what they want to discuss with the parents. Okay. So let me talk a little bit about the machine learning or the AI that's used here. So I mentioned that although,
Stephen, sorry.
Yeah, go ahead.
Do you have any data on if this is disclosable? How many cases, what did parents choose to do depending on what they heard?
Yes, so I can give you some examples, and obviously these couples, you know, we have to protect their anonymity, but I think the very first couple, or maybe one of the first couples that did this, they had a family history of breast cancer, but they were not BRCA carriers. And so they knew in their family tree, lots of aunts or grandmothers or whatever in that tree had breast cancer. And so they were very interested in screening against polygenic risk of breast cancer. And indeed, of the, I think, 5 embryos that we screened for them, one of them was an outlier, had 99th percentile breast cancer risk, and they chose not to use that one. So that's a sort of very kind of perfect use case, right? So it's very analogous to people who know they're carriers of a Mendelian condition like cystic fibrosis, and they just want to make sure they don't implant an embryo that passes that legacy down. In this case, now we've expanded it to polygenic risks. So, my uncle and grandfather died of heart attacks. It seems to run in my family. Is there a way that I can just make sure I don't implant an embryo that passes that polygenic risk down to the next generation? So we've had 2 use cases like that, one for type 1 diabetes and one for breast cancer so far. We have not had, despite the fact that the press always wants to talk about the fact that among the conditions that we can predict is intellectual disability. The press always wants to report that, oh, they're screening embryos for intelligence, but we've not had any, that's never happened actually in any of the cases that we've processed. Yes?
So today, like if you're pregnant, at the time of the blood test and do all these tests.
Yes.
Do you predict that you'll be able to do your population?
Yes, so there are 2 issues here. One is obviously, I hate to use the word abortion, but it's very different to say I have 10 embryos and I was only going to use 2 anyway, so the other 8 I was going to donate to research or whatever, have destroyed. And I just want to make sure those 2, the one that I use is the one that has, you know, doesn't have some health issue. That's quite different from I'm pregnant and I may want to terminate my pregnancy. So obviously NIPT, that's a much harder decision, right, for that person to make. Now in NIPT, what they're doing is that they're using the circulating DNA fragments, the extent to which one can inexpensively assemble a full genotype profile sufficient to run these predictors on is unclear to me, although there have been published papers which claim that you can actually get to that level of accuracy from NIPT. So all of this endpoint technology of the predictors can probably be applied eventually, maybe even today, to NIPT. Question?
So, has anyone used the height predictor for your?
That's a great question. So the model that we follow in all of our work is the disease model. So we are reporting on elevated disease risk. That is all we report on. The height predictor is good enough to rank order embryos. We've done some internal calculations and we could actually do a pretty good job of rank ordering embryos by eventual height, but we do not offer that. So the issue is that we, you know, obviously in our mind, there's a very important medical benefit to this kind of testing, and we're trying to elicit the least amount of societal outrage or response possible. So we don't, also, we can do many cosmetic traits. We can do eye color, hair color. We can do male pattern baldness. There are all kinds of highly heritable traits that we can predict outlier risk status for, but we don't actually do those, even though we have requests to do that kind of thing. Question?
So presumably the traits that predict breast cancer also predict other traits. But do you have a sense of like what those traits are? And, you know, an example of selecting against some other.
Yeah. So I think you're asking in a sense a question about pleiotropy, but pleiotropy for polygenic conditions. And so what is interesting is that if you look at the overlaps of the predictor SNPs that are activated for the roughly 20 different common disease conditions that we now have some handle on, there's not that much overlap. So they're broadly independent. And of course, medical epidemiologists would have already known if, you know, oh, people who get condition A are also likely to get condition B, and that would have already been known. But it's interesting to see it at a genetic level. So we can just see that those risks are basically uncorrelated at the genome level. So you had another question? Oh, yeah. Yes, sir.
Is there any?
Yeah, so some copy number and structural variation is tagged by SNPs, but by and large, it's not. So the next stage of this whole research program, broadly stated, is when we get to a critical mass of whole genomes with good phenotype data, obviously we'll be able to see like how important is copy number as a feature to be activated as a possible predictor, right, as a feature within a predictor. And we just don't know the answer to that question. I should emphasize that a lot of the benefit from whole genome analysis is coverage of rare variants. So those rare variants, unless they have very large effect sizes, are going to be difficult. The status of those variants is going to be very difficult to determine because you need consequently more statistical power. So in other words, if you have a variant that only 1 in 10,000 people have, and you have only 100,000 people in your sample, you only have 10 individuals who are carrying that variant. If that variant has a huge effect, of course, you can detect it. But if it has only a modest effect, like most of the SNPs that we're talking about here, you won't detect it. So. Okay, so here I was talking about sparse learning. So the idea is that typically we'll have 1 million or maybe 10 million after imputation features that we're looking at as possible things to activate in the predictor. And even the most complicated traits, so height is about 25,000 SNPs activated in the predictor, and for cognitive ability, it's maybe 30,000. So those are the most complicated. But that is a small fraction of the millions of potential SNPs which could have played a role a priori. And so the eventual thing that the learning algorithm is trying to learn is very sparse. If we think about it as a vector of effect sizes, only a very small subset of the entries of this vector are non-zero. And so there is a kind of mathematical framework, which in signals processing, like if we went over to the EAD electrical engineering department or computer science department, they might call it compressed sensing. There is a very strong mathematical framework around solving these kinds of sparse learning problems. And so actually the way I got into this field is that, you know, when you're a physicist, oftentimes, you know, because physics is a very mature field and the problems are very difficult to crack, oftentimes you're looking at other things and thinking, oh, could I possibly make some contribution in biology or in computer science or something like that? We're always, before we jump, we're always trying to do our own calculations to figure out like, well, is there something to be done there? Like if I wanted to try to solve one of these complex traits, how much data do I actually need? Are they anywhere near having enough data? So interestingly, before I actually got in this field, I actually used the theory that comes from this area to actually work through how much data one would really need to solve these kinds of problems. And believe it or not, they're actually fairly precise results, which allow you to predict a priori. If you know the heritability of a trait, and you know something about the distribution of variance, maybe from some early GWAS data, you can make a quantitative prediction for how much data you need to solve it. So 5 years ago, we published a paper based on some simulation work we had done, which I'll talk about later if we have time. We predicted that once you get a few 100,000 individuals with good height measurements, you'll be able to, quote, solve height. And in fact, that is exactly what we were able to do. So within a month of the UK Biobank releasing its 500,000 genomes, we had the predictor working. So you know, I'm aware that in biology, theory is kind of useless, whereas in physics, theory is the backbone of physics. But in biology, by and large, theory has not been useful, but this is an example where theory is actually very useful.
I just asked a quick question.
Yes.
As you get more and more genomes, are the predictors going to get more and more accurate, or is it kind of asymptotic?
Yeah, very good question. So for height, for height and common SNPs, we've hit that asymptote. For all of these disease conditions, in our most recent paper, we have projections for how much better the predictors will get as we get more cases. The limiting factor is just cases. So if anybody here knows Francis Collins or people at NIH, the biggest guaranteed bang for the buck for NIH is just to pay for genotyping of cases. So I can predict how much more power and how much better these nonlinear curves will be if you give me an extra 100,000 people with diabetes or an extra 100,000 people with breast cancer, and just let me do inexpensive genotyping on them. So, okay, so what are some open problems here? So this is what I just said, is that there's still a lot of room for material improvement of disease predictors. None of them are anywhere near what I would call solved. There's predicted heritability that we have not recovered yet in the case of disease predictors, although not so much for height. The number of cases used is typically only in this range. So, you know, again, if you could give me 100,000 people with condition X and allow me to genotype them, the returns are very high. This is an open problem. Most of the trained data comes from Europeans. The prediction power, and we've done kind of careful studies of this, falls off with genetic distance between the training group and the target group that you want to actually do prediction on. And one hypothesis for why this is that the correlational structure of the SNPs varies by ancestry. And so your predictor might have activated a SNP which is tagging a real causal variant, but is not actually the causal variant. And if the linkage between that tag SNP and the real causal variant is different in the target population, then your prediction quality falls off. And so one of the main problem I was saying to Fyodor before the talk, the main problem we're working on right now is at a kind of theoretical level is what is the best method to transport a predictor without losing statistical power that was trained in population A to another population, population B. And right before I was here, I was in Taiwan talking to the Taiwan Biobank, which is going to have, I think, 100,000 genomes ready for analysis later this year. So this is the kind of thing that we're working on. Okay, now I want to spend a little bit of time on the controversial trait. So this is data from a project called Project Talent, which was done by, I think, basically education researchers. And they psychometrically tested 100,000 American ninth graders. And so this axis is their mathematical ability, this is their verbal ability, and this is their spatial ability. And you can see that they don't fill out this cube uniformly. They actually lie on a kind of ellipse, and the ellipsoid. And the ellipsoid basically means that there are intercorrelations between math ability and verbal ability and spatial ability. So what's generally called general cognitive ability is actually just the coordinate along this major axis of the ellipse. They just take an individual, they project them onto this major axis, and then that coordinate is what people report as their, quote, IQ score or as their general cognitive ability score. And the best predictor that's been trained with about 1 million people correlates about 0.3 or 0.4 with actual IQ. So if I only have your SNP genotype, but I want to predict your IQ, I can predict it. It's a noisy predictor, and the correlation is about this. If you run through all the theory stuff that I was just describing, you can convince yourself you can get to a correlation of about 0.7 if we get more and better data. Now, this is an example of out-of-sample validation of the cognitive predictor. So,
Wait, I'm sorry.
yes.
So, forgive me for being devil's advocate.
Yeah.
You're making an assumption about narrow sensory ability.
Correct. So in getting to this number, so this is an actual reality. So this is something we do every day. to get to this, you are using a value of the narrow sense heritability, which is estimated from twin and adoption studies. And actually, there are some more sophisticated molecular methods now for estimating heritability, but they all agree. And that's where I get this number. Yeah, this number might turn out to be wrong. Okay, so this is an example of validation work on the existing predictors, which, as I said, are still noisy. These are 4 longitudinal social science cohorts. There's about 20,000 people between these 4 studies. These are people that have been followed since childhood. The researchers have information about their families, how they grew up, and then actually what they did in their lives. So the youngest people in these longitudinal cohorts, I think, are 40 or 50 years old, and the oldest people are like 80 or maybe even dead. So it's kind of like you have the whole life history of a person, and now you have their genotype. What can you predict if you blind yourself to everything except the genotype? What can you predict about their life outcomes? Now, in each of these, so this is in New Zealand. These 3 are American samples, and again, they were all born in different decades. So in some of these samples, the people were born sort of 30 years before the people in the other longitudinal study. In each case, there are 3 panels. One panel is people who are born in a high socioeconomic status family. These are people who are born in a low socioeconomic status family. These are people who are sort of middle class. And what you can see is the ability to predict upward or downward social mobility within class. So I have, for example, 2 siblings. I have their genotypes. I don't know anything else about the siblings. They were raised in the same family. They went to the same elementary school, high school, et cetera. But I predict brother 1 is going to make a lot more money than brother 2. Brother 1 is much more likely to have a PhD than brother 2 at age 50. Those are the kinds of predictions at a statistical level which we can make. And you can see there is real power in these predictions. So that's the current state of the art. And of course, this will get better. This will get much, much better if we get to this 0.7 level of correlation between the SNP predictor and the actual IQ. This one just came out in the last few months. This is a longitudinal study of 3,000 American high school kids. Longitudinal data again, so we know all the courses they took in high school, and we know what their final education education level. These are high polygenic score kids. These are low polygenic score kids. You can see important differences in life trajectories and life outcomes. When the researchers tried to control for the family socioeconomic status and the quality of the schools they were in, so they allow those as extra variables in the regression. They find they lose about 20% of the predictive power, but certainly not most of the predictive power. There's much more predictive power in the polygenic score. Yes. About 30,000 SNPs. So this is the thing that bothers people, but it is what it is. These are chicken breeds used in agriculture. The chicken that we ate for lunch today look like this. These birds were raised in the same way, fed the same food, and photographed at the same age. They are the product of selective breeding on the part of farmers. At some point around maybe the last 10 years, agricultural breeding of animals switched almost entirely to SNP-based predictors. So the milk that you drink, the chicken that you eat, the eggs that they lay, the chickens are produced, the animals are produced by artificial insemination, and the sperm is evaluated based on SNP genotyping and modeling, which estimates the quality of the genetics. And you can, if you had gone to a farm in 1957 and you looked at maybe 100,000 of these birds, you would not find a single one that looked like this. So directed breeding has moved the species in a very non-trivial way, maybe 10 standard deviations in a relatively short time. And so the IVF technologies that I'm talking about have the potential to do this kind of thing to humans in the coming generations. And so obviously it's something that we need to talk about as societies. The phenotype here isn't necessarily trying to become like Shaquille O'Neal. It may be, this may be the brain size or the brain effectiveness of individuals. Yes?
And that's just IVF.
Correct. Well, you guys are the next phase of all that stuff. So yes. Yes. So right now, it's not for people. There are a few people who, well, actually, if you're, for example, working at Facebook or Apple, and they, as a side benefit, as a health benefit, you have egg freezing. It's not unusual to have cases where they have 20 or 30 embryos that they're choosing from now. And that could go way up if, for example, stem cell technology allows us to take, for example, skin cells and make oocytes out of them. That's already done in mouse, I think. So those things plus CRISPR mean that all the stuff that I'm talking about is going to accelerate probably in the next decade. And so again, I think it's very important that we as a society talk about all these things before we decide what to do.
Have to edit 30,000 loci.
Yeah, so that's an interesting question. So imagine that, just to take a round number, 100 squared is 10,000. So imagine that you had a trait which was controlled by 10,000 variants. The standard deviation in the number of plus versus minuses is the square root of the total number. So you may only need to edit 100 to have an impactful, right, consequence, right? You don't need to edit all of them, but if you edit 100, that would be enough to do something. You guys will tell me whether that'll ever be feasible. So this is the main talk, and the other slides I have are more about like machine learning and phase transitions and high dimensional statistics. Is there anybody here that wants to hear those slides? Or I can just stop and we can talk more about these things, or I can actually go into the hardcore stuff. Hardcore. Okay. Oh, awesome. Awesome. Yay. I have these slides were put there for you. So yeah, that one.
So do you talk about privacy concerns when you talk about shipping everybody in the world?
Obviously, the laws regarding what can be done and what protections people have for their genetic information, those have to be thought through. One thing I will say is that, which I didn't say in the talk, but I should have said, is that if you think through what this means for insurance, we'll have a situation very soon where someone can go to a private company, get genotyped, and get all their risk scores. And then that's what economists call asymmetric information. So I can then go and buy a policy from an insurer, and they don't know anything about my risks. And so I think insurance companies will be pushed in the future either to ask you to submit some DNA with your application for health insurance, or we may be forced to go to single payer, because obviously in single payer, you're just pooling all the same risks. It's okay, right? But in the other case, insurance companies could easily be bankrupted by people who are at high risk for, say, early onset Alzheimer's, and they buy some long-term care package, and that one contract costs the insurance company $1 million loss. So I don't know exactly how this is going to play out, but the fact that we're suddenly now able to do these predictions, I think, is going to impact privacy as well as things like insurance.
And also like whether police have access to that.
Yeah, there's a whole nother thing which I won't talk about, but the way crimes are solved is changing radically also, because you can very easily from the crime scene get phenotypic description of the person, and then you can also match to a relative using a genomic database. So we're also very close to what I would call a genomic panopticon, where basically if you leave any DNA behind, they can find out a lot about you.
So all these models,
Yes.
are they transparent? Are they available? Not a person data, but actual model.
Yeah, the trained models. So most of the groups that publish these results, so there are two kinds of journals. For some journals, you're actually required to post the model itself as part of the paper. But even if you're not, typically in this space, if you email us or any other group, we'll send you the predictor. So yeah, we're all scientists. So yeah.
So as a quick follow-up, actually, to a strongly optimistic spin on this, you are identifying loci, alleles of which come together to create outlier phenotypes for disease susceptibility, presumably disease protection. Well,
Yes.
and so the age of CRISPR has brought us into the remarkable opportunity to sort of identify the genetic hierarchy, and then go from that to loci that encode things that we can actually drug for disease treatment.
Yes.
PCSK9 is a shining example, of course, but cardiovascular, it's relatively variant. If you have like 12 or 15 diseases there, and
Yes.
CRISPR is accelerating to a level where we'll be able, you mentioned the matrix of 10 by 10 or 100 by 100. We're pretty close to being able to model those kinds of cells. So.
Yes. So I didn't emphasize the low-risk outliers because it just turns out the way people in this field like to plot their risk data, the way we plot it doesn't really show what happens if you're percentile 1 instead of percentile 99. And so we actually know of individuals who have 1/10 or 1/20th the risk of a normal person. And so they have some protective polygenic aspect to their genome. And Katharison, who's the author of one of the papers that I showed you some figures from, has just left, I think, Harvard to start a company, which I think Google Ventures made a big investment in. And their goal is actually to CRISPR, I think it's CAD protective, coronary artery disease protective variants. I don't exactly know how this is going to work because I think they're going to have to inject you in your liver or something, or I don't know where, and who knows how often they're going to have to do it. I don't know, but it seems like very ambitious what they're thinking of, but it's definitely, people are thinking in that direction. So I just want to say a little bit about the modeling. So the most general model you could write here. So let G be the information from your genome, like a list of what variants you have at a million different locations. Effect sizes X and Z, these are linear or additive effect sizes. These are nonlinear ones, which involve gene-gene interactions. You're trying to solve for X and Z. You have the G information for a bunch of individuals, and you have the phenotype Y for a bunch of individuals. And so the whole process is just to invert this to get X and Z. And for reasons I could go into if people are interested, it actually was a priori plausible that the additive terms actually dominate. There's something called, for those of you who know a little bit about population genetics, there's a theorem called Fisher's fundamental theorem of natural selection. But anyway, so it was not surprising that the linear term actually dominates, but now that's been empirically validated. And so the simpler problem where you, as a first approximation, ignore the nonlinear interactions, you're just kind of solving this problem. And the sparsity that I was referring to is in this list X. This vector X will turn out to be sparse. So even in the most complex cases, you have over 10,000 variants activated, but maybe the list is 1 million SNPs long. Now, initially, this was an underdetermined problem because the number of possible features P was greater than the amount of data that you actually had. And this is exactly the problem for which something called compressed sensing or lasso or L1 penalized regression was invented for. And these are methods which have a strong prior towards sparsity. So what you want is an algorithm that is highly efficient, but it has a strong prior built in that it thinks the answer at the end is going to be very sparse. And so it has to be kind of cajoled to turn on an effect size. It has to have very strong statistical evidence before it turns on an effect size. And it's this class of algorithms that have been seen to be very effective in this domain. Here's a picture of the problem. So this is a list of the phenotypes of an individual. This is the gene, each row here is a genome of an individual. This is the effects vector, and only a few of the elements of this effects vector will turn out to be non-zero. Okay, there's a geometric reason why something called L1 penalization actually turns out to be efficient and effective in this context, but let me skip this slide. We end up using an objective function, which has both an L2 and an L1 term. It turns out this function is convex, so it's a very fast optimization. You're basically just kind of sliding downhill until you find the optimum. And by simulation, it turns out, so there are theoretical results. Sorry, skip that. There are theoretical results which tell you that the amount of data that you need scales as the number of non-zero effects, and then logarithmically with the number of features that you need to analyze. So this is a known result, and then the coefficient for the critical amount of data needs to be determined by some kind of simulation. And so that's what we did. So here's some results. There's a famous statistician, guy who does high dimensional statistics at Stanford named Donahoe. There's a phase transition named after him and his former student, I think, who's now at Oxford. Let me explain what this means. So in these graphs, what we've done is define some scaling variables, which are ratios. So this is the ratio of the sparsity of the trait to the amount of data that you have. This is the ratio between the amount of data that you have and the number of features that you need to examine. So as you get more data, rho gets smaller and delta gets larger. So you kind of move diagonally along on this phase diagram. And what's interesting is that what Donahoe and Tanner showed is that for this class of problems, there's a critical line. When you pass that critical line, you start getting very good performance of your algorithm. So when you're trying to do stuff here, you just don't have enough data, you can't do anything. But as soon as you pass this black line, you can start doing real stuff. And so what we showed is that the overall coefficient in this context leads to an estimate of about 30 times sparsity. So if you have a trait that has sparsity of order 10,000, then you need about data of about 300,000 in order to solve it, or in order to pass through this phase boundary. It's very interesting that using matrices which are formed out of human genomes, we get exactly this kind of phase diagram, the same phase diagram that Donahoe and Tanner talked about. So if you're a theoretician, this is kind of exciting stuff. But the practical implication of this is that we can make a priori estimates. Once we know some things about the genetic, little things about the genetic architecture, or preliminary things about the genetic architecture and about the heritability, then we can predict how much data we need to actually solve it and capture all that heritability in a predictor. And so this is an unusual situation in medical science where someone can actually go to NIH and say, if you give me this many dollars, I will build a really good predictor for disease condition X. And so hopefully that will get done. If it doesn't get done here, it's getting done elsewhere. So the UK understands this kind of thing. Taiwan understands this kind of thing. I'm not sure NIH understands this kind of thing. So let me stop there.
So with this here, going back to what you're finding with scores, it only seems like on the order of a million cases.
Yes. Yes. So the disease situation is a little different from, there are sort of two categories. One is a quantitative trait where everybody has a number assigned to them. And the other one is like 0-1. Like this person reached age 70 and didn't, or age 60 and didn't show any symptoms of this condition. And ones are people who actually have diabetes or had coronary artery disease. The analysis of those two different types of problems is slightly different, but in both cases, we can make predictions for how much data you need. I think that in both cases, the amount of data in the millions almost guarantees you that you're going to be able to capture all of this heritability.
So I heard that the diversity of populations says how healthy the species is. And has anybody looked into what's the benefit of like really the diversity? Often like population rather than like because if you
Yeah.
the predictor method is actually construct like perfect no diseases. Everyone is with that.
Yes. Right. I think I understand your question. So the issue is the monoculture question. So in agriculture, you might worry, like I breed or design these exquisitely optimized crops that grow under ideal conditions, grow like crazy and solve the world hunger problem. However, they may be very vulnerable because they're all genetically very similar to each other, and maybe I've eliminated some variation in that species that would have been useful against some pest or some disease that comes later. And so I don't know how to quantitatively analyze this because I don't really know what the future risks are going to be for the human species. But for sure that effect is there, right? In other words, if we're all clones of each other, we're all going to have the same weaknesses, right? So somewhere in the limit of no genetic diversity, it's kind of bad. But I don't know how far we are from not having enough genetic diversity to be robust species. My guess is we're quite far.
I have a question about your previous deck and the cohort study.
Can we go back?
Yeah, I mean, I don't know.
Oh, okay.
Go back.
All right.
So essentially what I understand you saying is that from these various cohorts and having their genomes, you're able to predict their various upward or downward mobility and social structure.
Oh, yes, that one, yes.
So how do you square that, I guess, with the recognition that that we already know that a major driving force of mobility and what we would consider success in life is heritable in the sense that it's based on a society's structure.
Yes.
And,
yes.
The question of the
yes. Great question. Great question.
Is it really your genes, or is it the fact that you are part of a group?
Great question. So all of those social science studies have a major confound, which is that you can say the following: you did well in life because your parents had money or were highly educated. And if you assume the transmission vector for that cause is the fact that the money was there and the schools were good, then you get to the sort of typical conclusion. However, you also have to factor in that the parents that are successful systematically had better genes. And we can measure that now. So what we're actually talking about now, what is at stake now, if you're a social scientist, is an understanding of the trade-off between these effects, which are real, right? There is a correlation between social status of parents and eventual social status of children. What is the transmission vector? Is the transmission vector better genetic luck that the parents already had and then passed on the plus variance to the kids? Or is it actually the better school? And what I showed you in the data there is that the school effect is only 20% of the genetic effect. In other words, if we try to predict social mobility using only the polygenic risk score, genetic score, and then I correct it by information about the family, social, and economic status, and the school quality, I only reduce the genetic effect by 20%. So predominantly, if these results hold, again, it's, this research is relatively new, it suggests that society is already stratified in a fairly meritocratic way. Now, that's a terrifying conclusion for most people to come to, but I'm not a social scientist, let them fight it out. But the PGS score is an important tool in answering that question of what is the causal vector of transmission. Does that answer your question?
Yeah. I would follow up on that. So
yeah.
In a racist country like ours, there are physical traits that cause oppression.
Yes.
So
yes.
How do you know that in the case you're just talking
yes.
about, that what you're not measuring is like melanin?
Yeah, so let me repeat. So I should have said this more carefully. All the results I'm showing you are for Europeans. So there are no black people in the study. There are no Asian people like me in these studies, okay? So there is no melanin issue unless it's maybe like you could say South Europeans are discriminated against because they have slightly darker skin than North Europeans. But we're not talking about race here. We're talking about within a population of white people in Iowa, can I predict who's upwardly, like even within family? So two brothers, full sibs, who's the quote smart one and going to go like this, and who's the quote not so smart one and going to do this? And can I tell that from the genome? And seems like the answer is yes.
So, in the dynamic presentation, really, see what the I guess my question is more than which is that when you operations define your traits, and you say that this is a trait that we're going to do that because that's what's available to us. We know that this technology is better person than someone who's right. So by you just choosing to like scratch that scientific itch, you then define intelligence for potentially someone else to use that technology. How do you think about it? How do you think about the new operation of intelligence or upward mobility, or whether or not we should be asking the question of monetary outcomes based on medicine, and if that's how we should measure society? Basically, what you're doing is you're enabling us to put these measurements on society, which is incredible.
Yes.
And it's like just mind-blowing. But just by doing that, you put measurements.
Now, remember at the beginning of the talk, I said, although I'm going to talk about like the UK health system and breast cancer and things like this, the strange attractor in the space of concepts for us to discuss is going to be embryos and intelligence, right? That's what always happens, okay? But just so you recall, most of the research is not about that, okay? But it's an important, but I understand this is an important societal issue that we have to think about. We have to understand what the science says and then make decisions about how society should be. Now, this construct of general cognitive ability is about 100 years old. So this intercorrelation structure of psychometric variables, if you go over to the psychology department, has been known for 100 years. It's only just that it happens to be a phenotype now that we can predict using SNPs, using genome. So we're not, to use the fancy term, reifying this trait. This trait existed in many, many textbooks, and it's actually used to filter who gets into UC Berkeley and Harvard already before any genomicists came on the scene. However, I understand that there's a powerful tool now available to you, which is that you can look at someone's genotype and say something about this quantity. Now that by itself, for people who are skeptics of this quantity, should be quite a shock, right? If you say, this is all socially constructed. Well, if it's socially constructed, how come I can predict it from your genome? Interesting question, right? But anyway, well, no, a lot of people don't believe that. A lot of people think this is totally socially constructed. Well, if I can do within I mean, if you're a social scientist, yes, what's the strongest possible thing that you could do within family prediction? Same environment, same shared environment, 2 kids raised in the same family, went to all the same schools. Brother is 1 year older than the other brother, but I can predict who's going up and who's going down. Pretty strong social science, empirical evidence. And that can be done now today. Could not be done 5 years ago, but can be done today. Yeah.
I share the concerns of what's been said here so far about social aspects, but there's another thing that's also concerning to me. And I think that's if we begin to reduce the concept of what determines health to being just genetic as well. So if we're not talking about cancers that may be caused by exposures to things that just happen to be more common among people with fewer resources, and, you know, that were just, it just troubles, my background is health, and so it troubles me a little to reduce as if we could just reduce it.
So I don't want to give the impression that these genetic predictors are ever 100% determinative. We're only identifying people who have more risk for something, not I know you will, although in some cases you can actually say you will get it, but in general we're just saying these people are at higher risk. Now again, the gold standard for this kind of study, if you think like, oh, you're really just picking up population stratification or some other thing you're picking up and it's not really an honest causal genetic effect. One thing is obviously the out of sample testing where you go to different environments and test the predictor, but the gold standard is always in family designs, within family design. So in other words, 2 brothers, same environment. One of them though for some reason had high risk for coronary artery disease and had a heart attack. The other one ate butter his whole life or whatever it is and had no trouble. So that's the gold standard and that is being done. So as this subject reaches its maturity, you'll be able to look up huge studies where they only looked at siblings raised together, and then they showed that the predictor actually differentiates outcomes or risk between those siblings. And so at that point, it's like, yeah, there could still be some environmental difference, maybe the genes made one kid like broccoli, and broccoli is really the causal vector. And so those things actually have to be looked at as possibilities. But I think the Occam's razor view of this is that, yes, we finally are able to predict phenotype to some degree from genotype.
So you had said that these models were constructed for white people, essentially, and so they are not good at predicting other races. To get models that predict other races, do you have to train it specifically on them, or can you just incorporate them?
This is the question we're exactly working on right now. So again, just to repeat, all the results I've shown you are data from European heritage populations, and we can exquisitely screen, like if someone has even like 1% non-European heritage, we take them out of the sample. So these are all Europeans, and all the validation is done on Europeans. Everything is Europeans here. However, I didn't show you these results, but we have results from taking these exact predictors, running it on a population of East Asians, South Asians, African Americans. So we have that, and we know the decay in predictive power as we move in genetic distance away from the training population. The big question is, I have, at cost of all this genotyping and all this stuff and analysis, I have used the statistical power from the European population to do something. Can I transport that statistical power? How much of it do I have to lose if I want to run it on East Asians? And that's an open question that we're trying to solve. We have developed algorithms which, given another sample of the target population, which maybe is not as big as the original training set, we think we're going to be able to preserve most of the predictive power, but we don't know the answer yet. In fact, that's why I was in Taiwan, is to get access to that data. So
that's all of us at our Polygenic course. Thanks, Stephen, for